So thanks Sarah and Vasily for inviting me, and indeed I will talk about why we benefit from being random. But before I start with this, I would actually like to briefly motivate how I myself grew interested into random numbers. Can I have something to move the slides? Yes. Thanks. So when I was 8 years old, like most children in my class, I was very interested in silico simulations of biological processes. Now, you can guess what in silico simulations those were. The year was 1996, and I really wanted a Tamagotchi. However, unfortunately, my parents were very much against electronic toys, so no Tamagotchi for me. Instead, I got access to my mother's PC and a book, Computer für Kinder, which featured some very interesting programming know-how, including if control structures, for loops, and some random codes to move faces around the screen and such. You can already guess what happened next. Combining the knowledge from the book and the computer, I kind of created my own Tamagotchi. The problem with this, though, was that the authors of Computer für Kinder had not deemed it necessary to include some kind of random number generator into their book. So this Tamagotchi, every 5 minutes, it would want to get fed. Every 10 minutes, it would get sick. And every other 10 minutes, it would want to play a game. And the game was really the most horrid thing ever, because it would ask you to guess a number between 1 and 10. And being the author of the game, I kind of knew the answer, so I guessed 9. And the Tamagotchi was very surprised. It was like, oh, that's actually correct. Can you guess another number? So yeah, I kind of provided. And this is how I grew very bored in computers. I then went to play in the forest and went on to become a biologist. But really, the lesson learned was that life without random numbers is very lame. 17 years, I think 17 years down the road, now I'm actually wondering whether life as such requires randomness. And I would like to start here with some illustrative example. Imagine you're a bunch of bacteria living in a very favorable environment. That's nice as long as the environment pertains. But once the environment changes, you're in trouble. So there are, of course, many strategies to tackle this. One of which is that you're many bacteria, and you can always have some of you switch into another state which is prepared for the different environment. You can also randomly switch back, but the important thing is always some part of your population is prepared for an environmental change and will therefore suffer in the environment which it's currently in. If then the environmental change occurs, your mates will cease to exist, or at least stop to grow, but you can proliferate, regenerate the population, and you would then again always make sure that some part of your population is in the other state, prepared for the adverse environment, so that if the environment switches again, the whole population doesn't suffer but continues to proliferate. And this is an evolutionary strategy that many bacteria actually employ. But more recently, it's also becoming clear that this plays a role in cancer. One used to think that the reason why cancer always comes back after chemotherapy is mostly genetic differences, that you always have some clones among your cancer cells which are genetically different and therefore prepared to take the chemotherapy. But in the last 2 years, a lot of even in vivo or experimental evidence has come out which shows that actually sometimes even the cells are all the same, but some just are in a state which usually doesn't allow them to grow so fast. But then if the chemotherapeutic comes, these cells would still be able to grow, to survive the chemotherapy, and then grow back the tumor after the chemotherapy has been taken off. So these kind of so-called bed hatching strategies are really important in many processes of life. And now let's look at why this is actually happening. So cells are actually random number generators. So cells are the thing I was looking for when I was 8 years old and trying to program a Tamagotchi. On the left hand, you see some bacterial cells which express various amounts of green and red fluorescent proteins. They are all genetically the same. They are all in the same environment. Nevertheless, their gene expression differs. Yeah? On the right-hand side, you see some embryonic stem cells, which are also in the same environment. They are also all genetically identical. Nevertheless, they differ very much in the expression of an important transcriptional regulator called NANOG. Maybe you've heard of it. So how and why does this actually happen? We've seen some why with the bacteria bed hatching. So let's go into how. You have some cells, so two cells, maybe a German cell in the left-hand corner, Brazilian cell in the right-hand corner, and they compete for making mRNA molecules, so for transcribing the genetic information. And by diffusion, the germ polymerase diffuses to the gene promoter and makes an mRNA molecule. The Brazilian polymerase maybe takes takes some more time to diffuse, yeah, but then in the end it will also make a molecule. And these sort of diffusive processes, while random, they will average out, so we will not get very large differences by this kind of mechanism. So the mechanism that's really at play is a different one. DNA exists in two states. It can be open and accessible for transcription, or it can be closed. And the switches between these two states, again, at the level of single cells, happen with the random kinetics. So if in the germ cell the chromatin is open and accessible, the polymerase will will transcribe the genetic code, produce RNA and ultimately protein many times. While at the same time, the closed chromatin in the Brazilian cell is not able to being transcribed, and only when it by chance opens up, a burst of expression can occur. But maybe this is relatively short, because then it closes again. So yeah, germ cells. Now you might actually think why, so we have how many genes? 20,000, right? 20,000 genes, so there's a burst of expression, one gene in one cell, and the other cell, another gene burst, so they are different, but why does that matter? I mean, it's just noise, we don't care. You have to know that all genes are interacting by regulatory interaction, especially regulatory genes. So if we have a burst of expression in a very central gene, some protein is made, then this will propagate along the genetic network because this gene regulates the expression of another gene. And depending on how the genetic network is wired, this can lead to a feedback and reinforce the expression of the first gene, which ultimately leads to a stable state that cannot be reverted by random processes. Therefore, changing the cell into a different state. So initially the clue was random, but then these random clues are reinforced to trigger stable switches. And this is, for example, important in development. So you know that we all start off as a single egg, a single cell, and then we need to break symmetry a couple of times. So we first divide between extra-embryonic and embryonic tissue, then the embryonic tissue needs to divide into the 3 germ layers. The germ layers need to divide into all the different cell types of our body. So there are many events in which symmetry is broken. And I don't want to go completely into the details of this. There are some events where this is actually extrinsically controlled. For example, the sperm entry site obviously induces some asymmetry. But by now it's known that in some other symmetry breaks, the first initial clue is random. So for example, the random expression level of this transcription factor called NANOG is then reinforced so that the cells would sort themselves by being green or red initially to then form the 2 germ layers. More down the road, and this is purely hypothetical, I think that these stochastic or random gene expression events are even important for many diseases. For example, for Parkinson's disease and other neurodegenerative disease, the current state of the research allows us to explain less than 10% using either genetic or environmental predictors. And considering that these cells are living for a very, very long time in the brain, I could conceive that just random transcriptional events or other events that occur by chance in a very low number of cells might ultimately drive a transition towards a disease state that then later spreads around the entire brain. But this has not yet been shown experimentally. So where's my own contribution in this? I develop and apply methods that allow us to observe the expression of, I don't want to say all, but at least many genes in individual cells. which then allows us to really look at what parts of the genome are more randomly expressed, what parts of the genome are more tightly controlled. And with this, I would like to quote, end by quoting two great scientists. The first one said that God does not play dice, but I think as biologists we don't really care, and instead we no longer play nice. Thanks.
Thank you, Lars. Any questions for Lars? Any random questions for Lars?
So how random is random?
It's more random than a Poisson. So the variance exceeds the mean.
Other random questions? No? I think it's because it was a great talk. Really.
Thank you.
All right. Lars, everybody.